Again, I'm Wes Henry with Capital City Beekeepers, as Ruth had told you. And without further ado, we do have our featured speaker, Cameron Jack from the University of Florida. So he's going to come up and he can tell you more about himself. And I know that you will enjoy the presentation that he has to give this morning. So help me welcome Cameron.
Hear me all right with this? All right. Hey, good morning.
Morning.
It's good to see you. Second time I've been to Kentucky. It's a little colder than the last time I was here. I came a few years ago for EAS. That was in Shepherdsville. So maybe some of you were there. Yeah, it's great to be back. So I know we're a little bit behind on schedule, and we could talk all day about Varroa, and we kind of are. If you looked at the program, we're going to talk a lot about Varroa today. So this talk that I'm going to give is really meant to be kind of more of an introduction. I'm going to try. I know we've got a mixed group of beekeepers here tonight or today that's going to have like some really early experiences, and you need to know about Varroa. And so we're going to cover some basic biology. I'm going to try to make it exciting because maybe at 9 in the morning, like a biology talk is not what everybody's like super excited to hear about. So we'll try to mix it up because you've got to control Varroa. I'll just tell you right from the beginning, we have to control Varroa or your bees are going to die. Everybody else here gets to talk about like the fun parts about beekeeping and all the, yeah, bees are so exciting. And like my job as a honeybee researcher that studies pests and pathogens is like, I just bum everybody out. It's like what I do professionally now. So I recently had a training and about how people learn, and there were basically 4 groups of learners, and you had to have certain things. Like one group needs to know what the presenter's credentials are, or else they're not going to listen. So just so you know, I have a PhD in entomology, so I study insects, right? And then there's another group of people that need to know, like they don't care about your credentials, they just want to know your experience. So I will say I've been around honeybees for most of my life. I'll tell you a little bit about that in a second, but I've been keeping bees myself for about 13 years now, so now we've got a little bit of credential that way. There is another group, happens to be the group that I fall into, that says, I don't really care, get the show on the road, or get going. And so some of you are already feeling that. But then, unfortunately for you and me, there's a 4th group that needs to know about me. So I'm going to talk just a couple minutes. I know we're going to be fast, but I'm just going to introduce myself a little bit here. So you notice that my first slide has nothing to do with bees. So my background, when I first started college, I went to a small liberal arts school in, it's called Southern Utah University. How many of you have heard of it? Of course not. No, 2. All right. Okay, we got a few. Yeah. So this is there was nobody in my university that was really doing research because it's just a small teaching college, and there just happened to be one person in the biology department that was really doing any kind of research, and they were doing surveys for the national parks in southern Utah. And so I was in a class, and I guess I knew that I liked science in high school, right? But as a high school student, it's like, what do you do with that? I guess I'd be a doctor? Like, what else is there, right? So I started down the pre-med track. I had my first class that had a cadaver lab, and I was like, nope, this is not where I am supposed to be at all. And then, so I had another class with this guy, and he was talking about his bat research, and so I just asked if I could go with him, and it turned out, like, all of a sudden it was like a light bulb. It was like, there are other kinds of science. Like, I can do other things than study people. And so that's where I got into it. And I will say I loved my bat research a lot. My wife didn't love it because I was gone all night, but it was super fun because it's like fishing at night. You just set up your nets, then I just come back and check my nets and pull out the bats. It was great. But I ended up going to, for my master's, once I started looking around, I ended up going to Oregon State University, go Beavs. That's like the wussiest mascot maybe of all time. But I was studying Nosema serrana. So this is a gut pathogen. If you have been around Tom Webster, you know a lot about Nosema, right? Because he is like the Nosema guru. Tom, I don't even know if you remember this, Tom, but I had a phone call with you as a young master's student, just because Ramesh Sagili was my advisor, and we just were talking, thinking through projects. So I've known Tom for a while, even though you probably don't know me, but I knew you for a long time because of all the Nosema work. But, so here's what would happen is I would go to my meetings and I'd be talking about my Nosema research, and then I'd get 100 hands that would go up, and then everybody would just ask me about Varroa. And so I was like, okay, well, I should probably go to, I wanted to go somewhere like where I could do a lot of Varroa research for my PhD. So I went to the University of Florida, and I won't say go Gators because I know that's could be a touchy subject, so I won't say that. And so I was doing my PhD work with Jamie Ellis there for mainly focusing on Varroa control. So a little bit about me, I have 4 daughters. I just spent 12 hours in a car with them, and so, you know, I'm not like super excited about it right now, but ask me later and then I'll tell you how cute my kids are, but maybe not right now. So I am originally from a small town in southern Nevada. My laser pointer, can't really see it, but I'm way up here. So about an hour outside the glitz and glam of Las Vegas, Nevada. And I can show this image in Florida, and there's like an audible gasp in the room. Like, oh my goodness, what happened? Like, where are all the trees? And the answer is there are no trees. So I just learned what green was later in life. But my grandpa was a beekeeper, so I grew up around honeybees and beekeeping. And I like to tell people that, like, I liked bees before it was cool, right? Because, like, when I was a kid, it was weird. And so my grandpa, you know, he was a sideliner. He was a high school principal that supplemented his income with bees. And so we kind of grew up around, you know, honeybees and honey production. But I think the effect that my grandpa had on me, probably more than I realized later, was that he was an educator, right? He wanted to share the love of beekeeping. And so, like I said, when I was a kid, it was weird. I remember one day where my grandpa, like, I went to my elementary school class, and my grandpa was in there, and I was like horrified. I could see some bee stuff out on the table. I was like, no, Grandpa, no. Don't tell, like, I've been trying to keep this a secret, right? And, but then he pulled off a cover for the observation hive, and then everyone was like, whoa, and I was like, yeah, that's my grandpa, right? Like, that's when it was like, oh, these are cool. And so it wasn't, like, it was kind of something that I've learned to love a little bit later in life. But now I am kind of following in this footstep of where I really like to teach, and I like to talk about bees, and I have a really cool job. I think I'm unique in the fact that I have like a primarily teaching focus, and I don't know that there are any other honeybee teachers that are like work for a university system really out there. There's a lot of that do extension, a lot that do research. There's not really very many that I don't know of anybody else that has like a significant or a majority of their teaching appointment. I was for a few years. I was 100% teaching, and so I've been creating a lot of courses. This year I'll finish all the courses that I ever want to build. This will be my 10th course, so I have 10 different honeybee classes, right? And think about that. Like at the University of Florida, I will have probably about 600 students a year that are taking some different beekeeping courses. Now, I know that most of them are not going to become beekeepers in their life, but they are voters, right? Like, so I think it's still a good thing to get some people introduced and learning about beekeeping issues and just kind of knowing how it all works. Okay, sorry for that third group that had to bear through that, but the fourth group needed that. So, all right. So now let's quickly talk about some Varroa. So Varroa, this is a parasitic mite. I like to point out to people because I get questions about this still all the time. My pointer doesn't quite seem to show up on that. But so Varroa jacobsoni, or you might see that name in the literature, and I still like maybe every once in a while I'll get emails about it. So I just want to point that out. Oh, we're dimming the light. Now you can see it a little bit better. So Varroa jacobsoni is what, or this was the name that was given. There is a mite called Varroa jacobsoni, but in the year 2000, there were some researchers that looked at the taxonomy or looked at the physical structures, the morphology, and they're like, actually, this is a different mite, and now it's aptly named Varroa destructor. But there's, we've been dealing with Varroa for so long. I mean, in the United States, we've been dealing with it for over 30 years, but in other locations, they've been dealing with it for even longer. And you'd think with all this time, we'd have some really good, solid solutions, but there's still so much to learn about this pesky mite. And part of the reason is that it's just so hard to study outside of the honeybee hive. Like inside the colony, I mean, we know that they're hard to kill, but then they're so hard to keep alive the moment we take them out of the hive. And so it's really slowed our ability to study this pest. So just as an example, one of the things that we've recently learned that you think, how did we not know that before, is that we've previously thought that these mites were primarily feeding on hemolymph, which is like the blood of honeybees. But then, kind of after some work with a lot of people, they found that actually these are feeding on fat. So it's just things like that. So this is kind of some of the basic research of what they did to determine actually these mites are not feeding on hemolymph, they're feeding on fat. So what it was basically somebody who was an eccologist, not even, he studies mites for a living, didn't really focus on honeybees from that angle, kind of looked at the mite's physiology and just physical features, were like, this doesn't look like it feeds on hemolymph. And then so there were some researchers from Maryland, so you probably have heard of Sammy Ramsey, and then Dennis Van Eagelsdorp, and then there were lots of groups of people that were working on this, but they basically would look at the mite. So this is a bee flash frozen in liquid nitrogen, and like, look, there's the mites tucked up under the sternites. So this is important. I want to make sure you see this, because there are a lot of people that look at their bees and be like, yep, I don't have mites. But they're actually really hard to spot when they're tucked up under the sternum. So just because you don't see them doesn't mean that they're not there, okay? And then what they would do, then they took some cross sections of bees, they break open the little bees, and then you can see like imprints of where the mites are going in and feeding. And if you cut cross sections, you see, oh, there's actually lots of fat tissues around where these mites are. Probably the most simple experiment that, again, I can't believe nobody did for such a long period of time, is that they fed bees a dye. They fed actually two different dyes. They fed them a dye that would stain their fat red, and they would fed them another dye that would stain their blood yellow, and then they put mites on them, and then they collected the mites, and then looked at the colors, and look at what color mainly showed up was red, right? There's still a little bit of yellow in there, so they're getting some hemolymph on them and in them, but they're primarily feeding on fat tissue. Now, what does that mean for you as a beekeeper? Doesn't really mean anything, right? At least not at this point. But what I wanted to let you know, the whole reason I'm telling you all this is because there are still some really basic biology things that we are learning about this mite. So a lot of work that still needs to be done. Now, the mite itself, the fact that they are feeding on hemolymph or feeding on fat instead of hemolymph, again, that's not really the problem. There's not like they're just totally draining the bee of all of its resources. That's not what causes the issues in the colony. It's actually the viruses that these mites vector and transmit that cause the problem. So, and it's not even necessarily that these mites are introducing new viruses into your honeybee colonies. Again, I told you, I like to bum people out. This is what I do. So bad news is that your bees are already riddled with viruses. Ooh, sorry, that's just what happens when you cram 50,000 ladies together in a box. Like, there's going to be lots of viruses that are going to be passed around and shared. The problem is then you add mites that can vector, which means they're like the viruses are replicating inside their bodies, and they're going from bee to bee to bee to bee to bee to bee. Now, those virus loads start to spread, to increase, and then the colony can come crashing down. So without viruses, mites would really be nothing more than just a nuisance. But it's, I mean, you've probably seen this. I mean, deformed wing virus is the virus that's most closely associated with Varroa. During the fall season, when you have high viral loads, you'll see those mites that start to have, or those bees that start to have shriveled wings. You'll see a lot of bees that kind of look dark and greasy. That's a really common sign of infection amongst insects in general, not just honeybees. Like insects that are infected with viruses tend to turn dark and greasy and hairless, and so you might see some bees like that that are pretty infected. So this is a kind of a close-up of the mite's anatomy. First thing you should notice: how many legs are we talking? More legs than 6, right? We got 8 legs. And that's how you know it is not an insect. This is an arachnid, right? But what is interesting to look at, and I think a lot of people don't get this close look. If you look at their body structure, I mean, where are their eyes? They don't have eyes, right? They're blind. Which probably most of you have not. I mean, I'm a scientist and a nerd, so I'm like playing with mites all the time, like trying to and it's crazy that they don't have eyes, because they can feel you and sense you and move around and move away from you. Like, so if I use paintbrushes to manipulate mites in the lab, and the mite can be sitting on the table and I'm coming in to scoop them, and then they start scurrying around. I'm like, you stupid thing, how did you know? How did you know that I wasn't there? Well, the way that they could really know is because look at the hair that's covered all over their bodies, right? So this hair is called setae, and bees, insects have this too. It's just they can feel the differences. These are like mechanoreceptors. So the differences in air pressure, just for me coming in for a landing to come scoop them up, they feel that, and then they sense their environment. Now they don't have eyes, and they don't have antennae, but they use their mouth parts. It's called the gnathosoma. They've got basically these feelers so that they can feel and taste and sense their environment, kind of use them like antennae. So the biologist in me thinks like, wow, these mites are pretty cool. And then, just so you know, so we're on the same page. Like the beekeeper in me, which is the majority of me, is like, oh yeah, we need to kill these mites. But just so you know, I kind of appreciate like how interesting they can really be. And they've got some other cool features that I wanted to point out. And part of this is so that we can know, it's important for us, if we're going to kill and control Varroa, we have to know a little bit about them. This is a close-up view of their mouth parts, their glycerin. So the way that this works is, you see that there's like some nubs up here, like these are their teeth, and then they kind of just poke in their mouth parts, and then they can kind of move their mouth parts like this, like little serrations, and then they can kind of just slurp up. That's the sound it makes too. If you listen really careful, you can hear that. So they scoop up some of that fat tissue, and they're pulling in. And another cool feature about mites that are in this whole family of mites is they've got this little organ right here. This is called a paratreme. Now I'm going to quiz you later on this, so remember that, paratreme. So paratremes are basically like little snorkels. So what they do is when they submerge themselves, and they can hide themselves in liquid, and they can still kind of breathe and hang out because of their paratreme. And we'll talk about that in a minute. And so the last thing, this is their feet, the suction cup feet. How many of you, when you were kids, wish you had suction cup feet that you could crawl on walls? Like, I thought about that all the time. So I couldn't be Spider-Man unless I got bit by some radioactive spider. So I had a better chance of being Spider-Man if I had cool suction cup feet. And the Varroa are living my dream with that. But we can also use that to our advantage if we can clog up those suction cup feet.
Could you reference the letter or the number that you're talking about? Because I just can't see where you're pointing.
Great, yeah, no problem. Yeah, you're right, you can't really see that. So the first one that I was talking about here has got that number 12 by it. So that's the mouth parts. So that's the ones that are coming in serrated. Then there's not a good number here. So I'm pointing at this kind of long organ that's pointing down right here. That's the paratreme, so that's that snorkel. And then over here, this all the way to the right is those suction cup feet. Got it? So I'll make sure I'll try to walk you through it when I see it. So, this kind of looks a little funky. So basically, this mite is all over the world. Now, this map is actually somewhat recent. It was just from last year that I pulled this map. We have a website, if you're interested. It's called worldhoneybeehealth.com, and you can find basically maps of all the pests and pathogens that infect honeybees all over the world. And we try to keep those updated kind of somewhat regularly. So notice, out of all the world, like the big beekeeping states, or the big beekeeping countries that are in red, right here, all have Varroa destructor present. Really, the only notable absence right there is Australia. Now, if you've been paying attention, or if you're tuned in, you've probably heard, actually, a few months ago, they found some Varroa in Australia. And if you've ever traveled to Australia, like, you know how intense they are about letting anything in or anything out of their, like, ecosystem, right? They're very super protective. So the whole country of Australia has been on lockdown, basically. And they've got, the Varroa showed up in New South Wales, which is kind of like the center of Australia, like middle east Australia. And they've been on lockdown for some time, trying to eradicate this mite. And they've been doing a pretty good job. I've heard that there's been a couple breaches, but for the most part, they were doing a pretty good job keeping it contained. So we're praying for our Australian brothers and sisters that they can keep this control, because, I mean, just for what we know, what it means to beekeeping, I mean, they've been able to keep it out. So we're hoping that they keep fighting the good fight. So where did Varroa come from anyways? So this mite has kind of evolved along with Apis cerana, the Asian honeybee, for thousands of years. And so what's interesting is that Apis cerana, doesn't really, beekeepers that manage Apis cerana, they don't really consider Varroa to be a pest. Like, there's nothing or it's a minor pest that they don't really have to do anything about, because these bees have seemed to kind of figure it out. They figure it out with a few different mechanisms. Interestingly, Apis cerana, they are just aggressive, right? Like, they do not mess around with this mite. When this mite is on them, they won't stop moving or shaking until they get that mite off, they bite it and kill it. So people have been trying to breed Apis mellifera for this hygienic behavior, where they're more likely to get this mite off of themselves. But something that's also really interesting, and we still haven't necessarily pinpointed the exact mechanism for this, but they just find that Varroa don't really reproduce inside Apis cerana brood cells, and I'll talk about the reproduction in just a moment. But they don't; they're only reproducing in drone cells, and there's a unique kind of behavior, unique biology related to Apis cerana, where even when they're reproducing inside of the drone cells, sometimes the drones will just die. And the workers are smart enough to not open up that cell and pull out that dead drone, and basically they call it entombing. So basically the mites get stuck down in there and they can't get free and they just die. So there's these different mechanisms that have prevented, or that just have kind of balanced out probably over time with Apis cerana and Varroa. But Apis mellifera doesn't have those same mechanisms. That's why it's such a pest. And the way that we were introduced to this in the first place was once we started moving Apis mellifera kind of outside of its native range, when we beekeepers started moving it over into Russia and China. The first incidents where somebody reported and published, hey, we're seeing this mite on Apis mellifera, was 1952 what was then the USSR, right? And then we saw it later pop up in Japan and China, and then, so that was in the 50s, and then in the 60s, oh, hey, look, it's in Europe, oh no, in Bulgaria, and then in the late 70s, it showed up in Germany, and from there, boom, it just like started hitting everybody around the world. And so we were kind of actually one of the last holdouts of major beekeeping countries, right, where we didn't get it until 1987. But so we've been dealing for it for 30 years. In Europe, they've been dealing with it for 40, 50 years, right? So there's been a lot of work that has been done related to Varroa around the world. So part of that spread that make Varroa able to spread like wildfire, that's what I'm trying to say. So it's partly because of the nature of the bee, and then some of it is just to the nature of the beekeepers, okay? So, I mean, whenever you have bees going into other colonies, like through robbing or through drifting, you're going to going to be like sharing basically whatever pests or pathogens are on those bees. But then, as beekeepers, what we do is, we can strengthen a colony. That's why we're giving new advice to new beekeepers. We don't usually say start with one colony. We say start with 2, right? So you can balance them out. You can strengthen them. But every time you're strengthening a colony and you're moving frames of brood around, you're going to be moving Varroa around as well. And then just due to the migratory nature of commercial beekeeping, that just has made it, spread so quickly. So this is Varroa can spread in an area in a hurry. Okay, so in order for us to better understand controlling Varroa, we also need to know basically how they reproduce, because we can use some of this to our advantage later on. So let's go from left to right. So we've got a queen lays an egg, right? And then we've got our larva that's developing, and then this first mite that is going to enter into a cell is called a foundress. And this foundress is always going to be a female. This mite, what she will do is if she just hangs out on top of the larva, the other bees are going to see her and then just try to get her out, right? So she's sneaky enough that she will kind of wedge herself underneath the larva and kind of submerge herself down in the brood food. So now this is where that organ comes into handy. Who can remember? What was it called? Paratreme. Yeah, I heard a couple people say, yeah. So this is that organ comes into play, right? The paratreme kind of allows them to submerge themselves. They can breathe, and then once that cell is capped over and that mite or that larva eats all the brood food, now that mite is free to move about the cabin, and she can then feel it's called a cell. That was a joke. It's not very funny, Cameron. Okay, so she feeds on she'll puncture the larva and then just use that one location to just feed and pull that fat and hemolymph. And then what she'll do is she'll lay an egg. So you can't really see if you can see that small little dot. That's an egg. So she'll lay an egg about every 30 hours. So about once a day, she's just pumping out egg. Now, this all their reproduction and all their mating all happens in this short period of time. From the time that this cell is capped, if we're talking about workers, it's capped to the time that they emerge out as an adult is only 12 days. So this is a lot that's happening in a short amount of time. So that first egg that hatches will become a male. And then every subsequent egg after that is usually a female. And so what will happen is that male son will then mate with all the male daughters. Sorry, that doesn't make any sense. So it will mate with all of the female sisters. That's what I meant to say. So that the male son, female sisters, they will mate, and that's the only mating that they will do for the rest of their life. And so, which is really interesting, because you just think, like, that's a tremendous amount of inbreeding, right? But they seem to have it figured out, because they're not turning blue or growing extra limbs or anything like that, right? So, but in that short amount of time, again, so this worker now emerges, and what you will have, you won't usually see the males. They will stay behind and die. And there might be some females that are not fully mature. They're still kind of soft-bodied, and so they will stay behind in the cell and die as well. But in this short amount of time, this is a figure to kind of show how this works. So we're looking at workers. So again, this is a 12-day period from the time the cell is capped to the time that emerges. You have one female that goes in, that's the foundress, and then when she comes out, we've got 2 females that are mated, mature, ready to go, right? But also, you can have, this foundress can have, up to like 5 to 7 reproductive cycles, okay? So the mite populations can grow in a hurry. Now, if a lot of you already know this, but when you have a lot of drones inside of your colony, you can also see those mite numbers take off in a hurry, and it's partly because these mites are really partial to drones for reproduction, to drone brood. So there's probably several reasons. I just imagine drones are bigger and juicier, but probably more importantly is that there's just bigger dividends for reproducing a drone brood because they have 3 extra days of development. So this is actually a 15-day period from the time this cell is capped to the time that an adult drone emerges. And so those 3 days pay big dividends, right? You've got the foundress that goes in, and now you've got 5 fully mature females that are ready to go. So biologically, it makes a lot of sense for mites to try to reproduce in drone cells. This is a quick picture of what they look like. I took this from Zachary Huang, who's a missionary, Michigan State University. This is what the males look like. Again, they're kind of pink, they're round. You don't really see them unless you're looking for them by like pulling out pupa, like honeybee pupa or something. And then this is what they look like, the females look like before they're fully sclerotized. So when they merge out, they look a little bit soft, but they'll harden up really quick. So this is kind of what the mature females look like. All right, so we'll get into some control in just a minute. I already know that I'm kind of getting behind, but I'll talk a lot about controls in some of my other talks as well. So we do have, this is important to know, is kind of the development cycle of Varroa and kind of the seasonal growth. So this blue line, let's start here. This is our bee population. I took this figure from the Tools for Varroa Management, which is a really useful guide. But you know, the honeybees start in a dormant phase. As the temperature increases, we kind of reach our peak population in around early summer, and then once we're kind of back into late summer and fall, our honeybee population starts declining. And now, because we know that our mites their biology is so closely tied to the honeybee's biology, right? So we see that this orange line is our mite population. So you see how it starts low and then it grows as the honeybee population grows. But then see, this is where it becomes extra tricky. It's kind of in this late summer, fall season when our honeybees are our honeybee populations are declining, but there's still enough brood inside the hive for our Varroa to continue to reproduce. And so their populations are going to continue to grow. So it's often in this summer and fall where we see really high virus levels. And then that's when the colonies come crashing down because there's such an overwhelming amount of mites. And that's when it becomes a major problem. So, as part of my message that I feel like that I go around preaching all over is that it's so important to actually know what your mite populations are. Now, I get a lot of pushback, and they're like, Cameron, we know we're already going to have to treat. So why in the world do we need to spend the time to actually monitor and know what our mite populations are if we know we're already going to have to treat? Which is a fair question. But what it really comes down to is that you don't know if what you are using as a treatment is actually effective unless you check, right? So in order to check, you have to know the before and after. I love, talking with beekeepers and going all over and working with beekeepers, and I get probably an email, maybe like once every couple weeks or so, of somebody or somewhere in the world that is solved it, right? Like they've got the answer to all our Varroa problems. That all you have to do is this, right? But almost every time, nobody shows me their before and after mite counts. So I always tell them, it's like I don't know if what you're saying is true unless you actually have before and after treatment numbers, right, of your mite populations. Have you ever heard the saying, "In God we trust, all others need to bring data," right? So this is your opportunity to get that data. You need to monitor for Varroa. Really quick on this is that your mite populations are going to change throughout the year, even if you're treating, and so you kind of do need to know where they're at, and at certain times of the year. So if we start at the top, like in this table where it says dormant with brood, what's acceptable is about less than 1%. We usually measure this in the number of mites per 100 bees. So we can say, one mite per 100 bee would be 1%, right? So if you have about 1%, you're usually okay when you don't have, or when you have some brood, but your colonies are kind of dormant. Let's go population increase. When we're in the spring and the summer, when our bees are increasing, you can get away with a little bit more. Once you get to about 1 to 3%, that's when you kind of need to know to really be doing something. When your populations are decreasing, around 2 to 3%, same kind of thing. So generally speaking, you'll often hear people put that line at about if you got more than 3 mites per 100 bees, then that's when you've got a problem. Now, I will say a lot of that depends on what you're going to do in terms of treatment and what your plans are for those bees. And we can talk about that a little bit later, but generally speaking, a lot of people put that at 3 mites per 100 bees. All right. So I told you a minute ago that a lot of people will just use visual inspection, and this is kind of what I just call the lazy beekeeping side of it. It's like when you pull out and just look and be like, nope, they look good, I don't see any mites. But I will tell you just, I know we're running low, but just a quick story, just to illustrate how ineffective this is I have the exact opposite problem of all beekeepers in the world, where I cannot get enough mites. And because I need them for laboratory work, right? And so we have to have these colonies that I have to manage in a certain way so that they are just bubbling with mites. It is very sad. You can feel bad about it if you want to. So it is sad. Like I have to have lots of mites. So I had been managing these colonies for a while, and this was part of my PhD work a few years ago. And I pulled out a frame, and I was expecting to get like hundreds of mites just crawling all over. And I looked at the frame, and I was so sad. I was like, I don't see a single mite on these bees. And I was like, well, I'm like, this is going to ruin the study. I had already set up all these cages and vials and stuff for the lab for all these tests that I was going to do. So I was like, well, I'm just going to collect them anyways, just to see, right? So I'll collect some bees, and then I went inside, and I started shaking off the mites, and I got about 60 mites per 100 bees, right? And I did not see them, okay? And I don't want to brag, but I got 20/20, and at least I did a few years ago. And so, and I didn't see them. So my whole point in telling you this is like, it's really hard to spot mites. I mean, when you do see them and they're hanging out on the thorax and you are seeing a lot of mites, you got a problem, okay? That's when your mite numbers are already pretty bad, or you just got really lucky. So I won't get into this super hard. I mean, there are a few different ways to monitor your mites. The powdered sugar shake is not my favorite, but it's better than nothing. And it does kind of allow you to get some idea, ballpark, of where your mites are at. You don't have to be in beekeeping to know that, bees probably don't want to be in a jar. That's something that's like really intimidating for like new beekeepers, and so they just don't do it. But I promise you, there are ways to do this without causing much problem. I almost never get stung when I'm doing monitoring, if you do it right. So that's another talk for another time, but do work on that, right? And so the monitoring strategy of choice for me is going to be alcohol washes. Part of that is just because I'm a researcher and I just need to know, I want to know the exact number. You are going to sacrifice some bees, about 300 bees, but it is a small price to pay, in my opinion, of making sure that I have an accurate picture of the health of my bees. And so, what you're doing, I have a honey sieve that I just dedicate to my mite washes. And then it allows me to kind of rinse I can pour out these bees after I shake them in alcohol for a while. I can pour them out, and I can rinse them under the sink, and then I can separate the bees, I can count them, and then I can count the mites that collect at the bottom. So it's not that difficult to do, I promise you. Just got to do it. All right. So, Philip, you're going to have to tell me and give me the cut sign when you just need me to start talking, because I can go all day. But you stop me when you need me to. Just throw something at me. So one of the things, like I said, I've got other talks later today where we're going to talk about Varroa control, and I know some other people are going to be talking about that. But we do have some good things going for us in terms of Varroa control. I mean, if you've seen this pyramid before, this is kind of like this integrated pest management framework that is often used when we talk about controlling some kind of a pest, and because it's not all chemicals. Notice that red top is the chemical treatment. So there are some other steps that you should be, trying out and employing before you get to that chemical control. I have recently Jamie Ellis and I wrote an article. This is free online. Anybody can look this up and read it, where we basically tried to summarize like the 50 years or plus of Varroa research and kind of put it into this IPM framework, where we were kind of talking about what was effective, what was not effective. And so you should definitely check it out if you're interested. It's a big read, but there's a lot of good information in there. All right, so again, I'm going to go quick, so know we're running low. So prevention, you might think that was, notice that was the bottom. I'll go back for a second. This is the very bottom of the pyramid, and you are already thinking, Cameron, we know we got Varroa, that ship has sailed, and yes, you're likely right. But just so you know, I mean, there are some things you can do to kind of minimize the amount of mites that are coming back into your apiaries. So for one of them, I think is actually an important part of beekeeping is controlling swarming. Now, I know there's some people that will argue against me, like swarming is a natural part of the mite or the honeybee's biology, which is true. However, as beekeepers, and especially if you have any kind of neighbors nearby, I mean, it's just not a good idea to let your bees just go out there for a couple of reasons. Because one, your neighbors will hate you very soon. But two, you know, the swarms kind of in the area will kind of serve as reservoirs for mites because they're not being treated, they're not being controlled, and so mites are just going to show up, and then your bees are going to kind of drift and mingle with their bees, and those mites are going to come back in. There are some people that will treat packages as they come in to their apiary. I don't personally do that, but some people do, and that's just an example of prevention. So we move up to cultural controls. Cultural controls are basically, a cultural control means you're changing the environment to make it less suitable for the pest. And so one of the ways that we can do that, an example of cultural control would be to breed hygienic bees. So what we're looking at is in the top right corner, you see how those bees are pulling out a pupa and there are mites on that pupa, is because those bees are detecting that there is some mites going on inside that cell. And so they're opening that cell and they're pulling it out, and then it kind of disrupts that reproductive process. Did you have a question?
What's the reason why we cannot or don't want to use Serena if
Good question. So why don't we just use Serena that are already able to do this? Well, again, it's that we don't really want to bring in another organism that is outside of its native range, because then there will also have more problems. And really, the reason they brought Apis mellifera back east throughout Asia in the first place was because Apis mellifera is such a good honey producer and is such a productive bees, where Apis cerana is not. If you have Apis cerana, you will be more of a bee haver than a beekeeper, because for the most part, they're pretty nasty. This summer I was in Thailand, and I got to, I led a study abroad class with students, and we got to go see all sorts of different honeybees, and it was a blast. Especially if you're like a bee nerd like we are. Like I said, yeah, like I got super excited about every time. But Apis cerana kind of wore me pretty thin pretty fast, because every hive we went into, we were like, whoo, these bees do not like us. So there's some reasons why we necessarily wouldn't want Apis cerana. And we can't, they're two different species, and so we can't really, I get that question a lot, why don't we just breed Apis mellifera with Apis cerana? But it doesn't work. You're going to get a mule if you breed them together, right? So another thing that would be like a cultural control that would be changing the environment is that like you could cause a break in the brood cycle. So some people have done different research on this. I've done some work on it, which I'll share later this afternoon, that where we didn't have like really great success with it. But the idea is if you can break the brood cycle by caging your queen for like 3 weeks, then think about, I mean, that completely stops the Varroa reproduction inside your colony, which could make them, you know, easier targets for other kinds of treatments. All right, mechanical. So mechanical basically means that we are doing something physical to that hive, and physically either removing or physically killing them in some way. That's what mechanical control means. So some examples of mechanical control could be if you recognize this green frame over here, this would be like a drone foundation frame. So drone trapping actually can be really effective at removing large portions of mites from the colony at a time. However, it is quite difficult to do. I mean, it just takes good note-taking, and the stars kind of have to align to some degree to really have enough drones in the colony to really make this work efficiently. But it's a non-chemical method that works great if you can make it work. And you have to be taking good notes, like I said, because what would happen if I had, I was trying to do a mite control, and I have a drone foundation frame, so I got lots of drones, but I don't remove that frame in time. well, what have I just done? I've detonated a mite bomb in my own colony, right? It's just caused so much more issues for me. So not something that it's something that you need to do if you're going to be diligent. There's kind of a newer, it's not new, I mean the idea has been around for a long time of hyperthermia, so the opposite of hypothermia. Hyperthermia means we're making it hot. And so there's a temperature, it's about 105 degrees Fahrenheit that starts to kill mites, but it has to be maintained for a long time. However, at like 111 degrees Fahrenheit is when it's going to start killing all your larvae. And so it's not, I'm not usually an adapter of new technologies quickly. Like I was probably the last person in my age group to like get a cell phone because I just like, oh, I just don't do like new technologies early. So I'm kind of waiting on hyperthermia for like everybody else to like work it out before I go into it. But I mean, the idea is plausible. It can work. I'm not endorsing it in any way. I'm just saying the idea does work. It's just hard to do. You'd have to have good equipment to really do it well. All right, biological control. So biological controls mean we are using nature, right? Like we're using another organism to control this organism. So there, people have been looking at different biocontrols for Varroa for a while, and unfortunately, like it doesn't usually work. There's a lot of things that work well in the lab, but then when you take them to the field, it's not so simple. So for instance, like this picture here is a pseudoscorpion on the upper right. Pseudoscorpions have been observed to like eat Varroa, and you put them in a petri dish together, and maybe that would warm your soul a little bit, like to just see the Varroa running in terror around while this pseudoscorpion is chasing them with the pinchers and gets them and eats them. The problem is when you take a bunch of pseudoscorpions, you put them inside the hive, you know what's easier to catch than mites is eggs that are just sitting there, right? So what they've had some videos that you can find on YouTube where you've got like pseudoscorpions that are just following the queen. The queen's laying and boop, 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 and then the pseudoscorpions are just following her in and out of the cells, just eating the eggs. So it doesn't necessarily work very well. There's been some entomopathogen fungi that like work again, like you spray it and the fungi will like develop in the mites, but they don't really spread everywhere else. You need to zip it. Okay, I'm getting the hook. Okay, so last thing I'll say, so chemical control. So chemical controls are really where the majority of us have landed, okay? Just because this is where it's been the most effective. But there are other options, there are things that we need to explore and ways that we need to kind of integrate them differently. So I'm going to give a talk later this morning about some different chemical controls, both synthetic chemicals and non-synthetic chemicals that we have tested to kind of give us a view. And so if you're interested in that, and then I'll give you some more information about that for now. So the last thing I'll say is just like Varroa is super important. You need to do something about it, or else your colonies are going to suffer. So good luck. There are ways to do it. Good luck. Learn about some today, and happy beekeeping.